Hello, guys. It's Wednesday, September 9th. And can you hear me? Yes, good,
Yes.
good. So today we should finish chapter 3, amino acids, peptides, and proteins. So we spent quite a bit of time talking about amino acids, and we started talking about peptides. So we're gonna continue on with peptides. So as we know, peptides are not really amino acids, they're not really proteins. Let me start sharing my screen with you. Now, don't forget the homework is due on Monday. So there are quite a few questions there, but as I mentioned to you last time, many of those questions are kind of quiz type questions, where basically you just have to pick the right answer. Okay, let's see. Share screen. Okay. All right, so peptides. So as we talked about, these are small proteins, right? 10 kilodaltons, so less than 10,000 in molecular weight, right? So this is just roughly. Now, people refer to some of the peptides which are heavier. They still refer to them as peptides. Many say that insulin is a protein, but it's actually based on this rule, it's a peptide. So this is just roughly. So the way we're going to make these peptides is by removing a molecule of water, right, from the carboxylic acid OH from one amino acid and hydrogen from the amino group of another amino acid. Remove molecule of water, and you get the amide group, amide. So we know that from organic chemistry. And I told you last time about esters a little bit. So there's a reason why nature chose amides, right? So amides are quite stable. Without enzymatic catalysis, amides are quite resistant to hydrolysis, right? So, and the reason for that is this resonance stabilization, right? So nitrogen donates electrons into the carbonyl group, and the carbonyl oxygen pulls electrons towards itself. Now, the same effect in principle should be possible in esters, right? If you have an ester like that, there's a lone pair. There's a lone pair that can do exactly the same. But yet, if you try to hydrolyze this with water, it results in a fast degradation, fast hydrolysis. Fast hydrolysis. Now, amides are characterized by slow hydrolysis. But as I mentioned, there are enzymes in the body, right, in cells, which can break down the amides quite efficiently. But without those enzymes, hydrolysis is very slow. And since you're attending this lecture, I'm going to reveal a secret. I'm going to put something like this on the test. On our first test, I'm going to actually ask you why the amides hydrolyze slowly and why the esters hydrolyze fast. So here you have to apply your organic chemistry reasoning and explain to me why this resonance structure that comes from the amides stabilizes the amide more than the same resonance structure that comes from the ester. So it has something to do with the nature of nitrogen versus oxygen, right? So think about it. And so be prepared to answer something like this on a test. All right. Now you have a number of amino acids, you put them in a chain, right? So it's a small peptide. Now this is 1, 2, 3, 4, 5. 5 amino acids, so we refer to this as pentapeptide. Pentapeptide, 5 amino acids. And by convention, we start with the amino terminus on the left, right? Will always be on the left. So don't get confused. And the carboxyl terminus will be on the right. And as far as the R group, you can put this on top, you can put it on the bottom, wherever you want to put it, it doesn't matter. Okay, so this is how we're gonna draw peptides. And we're gonna name this, of course, there are 3 ways to do it. We learned this already, right? One is serine, glycine, tyrosine, alanine, leucine. So 5 amino acids. Another one using 3-letter abbreviations, right? And the last one is using 1-letter abbreviations. So there are 5 amino acids. And as I mentioned to you, this is primarily used in computer modeling and bioinformatics, where basically you have to deal with large protein structures quite efficiently with and spend as the minimum amount of time on defining the primary sequence.
I have a question.
Yeah, ask me.
I don't know if it was obvious, but what does the N terminal mean?
N-terminus? I mean the terminus.
Oh, okay. Right. No, yeah, I get it.
Yeah.
Yeah.
So this is N-terminus, so left. So one way you can say amino terminus, another way you can say N-terminus. 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 And then what's this going to be called?
The C terminus?
C terminus, exactly. Same way. If that's N, this is C. C terminus. All right, good catch.
Got it.
All right, so yeah, sure. So we talked about this a little bit, right? Last time I mentioned about insulin. Remember, again, insulin, even though the molecular weight is less than 10,000, some people refer to insulin as a protein, right? But it's actually, by our definition, should be a peptide. Oxytocin induces uterine contraction, sex peptide in insects. There's a pain mediation by a small neuropeptide, antibiotics, now polymyxin B, right? Very important antibiotic. It's basically, it's a cyclic polypeptide. It has a basically highly effective against gram-negative bacteria. Now, if you have taken some microbiology, or you will take microbiology, you will learn that the bacteria is divided into gram-negative, gram-positive, depending on their structure. But gram-negative bacteria are really bad bugs, and they're what's referred to as hospital-acquired infections, right? So now it's all about COVID, right? So our number one worry is COVID. but it doesn't mean that the infections, bacterial infections, which are resistant to antibiotics, they have not gone away, they're still around. And many gram-negative bacteria are resistant to virtually any antibiotic we have right now. And polymyxin B is still the one that actually still works against these hospital-acquired multi-drug resistant MDR, multi-drug resistant bacteria. So polymyxin B still works against them. And that's a peptide, right? So very useful. And many peptides are toxins. Toxins from mushrooms, snails, scorpions. So, all right. So these are peptides. Any questions about peptides? Obviously, they're between amino acids and proteins. They don't adopt as extensive three-dimensional structures as proteins, and so they're not as efficient in regulation, for example, right? So the functions may not be as well regulated as with large proteins that have lots of subunits, that have lots of sites, which can have specific functions. So, so proteins and polypeptides. Now, I already mentioned to you that the term polypeptide, I'm still not quite clear what exactly this means. I guess in this text, they refer to polypeptides as large peptides, in other words, proteins, right? So that's what we're going to do from now on. Polypeptides and proteins are going to be interchangeable. So if you tell me polypeptide, I assume it's a protein. I, by myself, I will not be using this term polypeptide because I would like to stick to something that is universally completely understood and accepted, proteins. So proteins can, in addition to having the sequences of amino acids, they can have other components. And for example, cofactors. So when somebody says an enzyme cofactor, usually people mean some kind of metal ion. Metal ion, so many people take mineral supplements, dietary supplements, right? Take magnesium, zinc, manganese, calcium, anything, iron. So basically many enzymes utilize metal ions for their catalysis. And so those are referred to as cofactors and usually inorganic, which means metal ions. Now, coenzymes are usually small organic molecules. And usually they're present, for example, one of the most common ones is nicotine adenine dinucleotide, NAD, or NADH. We will also learn about this. NADH, so NAD plus is the oxidized form. NADH is a reduced form. So what this coenzyme does, it helps the protein, the enzyme, such as lactate dehydrogenase. It helps perform reduction oxidation. So these will be, in most cases, these are non-covalently attached, right? So this is associated with the protein. But there are other components, such as prosthetic groups, which are covalently attached. And one of the most famous one is heme in myoglobin and hemoglobin. So chapter 5 deals with oxygen transport, and we'll talk about that in great detail, and we'll look at the structure of the heme and we'll draw it. We'll draw the heme, so it's going to be called the prosthetic group. And there are other modifications. So it's, again, not just amino acids, not just 20 amino acids. There are other groups, cofactors, coenzymes. Who remembers what post-translational modifications means? Anybody remembers? Post-translational modifications.
Like splicing the mRNA. Is that kind of the right idea?
Well, with translation, so translation would be synthesis of proteins on ribosomes, correct?
Oh, that was transcription. All right.
Yeah. So post-translational, that means after the protein has been synthesized already. After the protein is synthesized, then there are enzymes that can perform other chemistry on these enzymes. So we talked about, for example, hydroxyproline. We talked about 5-hydroxylysine, right? So there are enzymes, oxidases, which introduce hydroxy group on these amino acids, for example. So these are examples of post-translational modifications. So when you think of proteins, don't just limit yourself to the 20 amino acids. Proteins are much more complex with lots of other components and chemical modifications. So proteins vary in size quite significantly. So I mentioned to you 10,000 is a rough estimate. And so here we'll start with cytochrome C, which is close to 10,000, just above 10,000. 100 amino acids, then we increase the molecular weight, some various proteins, and you can see they can be as large as Titan. Titan has the molecular weight of 3 million, right? It's a huge protein with with 27,000 amino acids. So there is also another thing to consider. There are a number of polypeptide chains, right? So it can be, Titan is one long protein, but one chain only, right? It starts with one amino acid and ends on the other end with one amino acid, nothing else. But there are other proteins which are what's known as multi-subunit proteins, multi-subunit. And if these subunits can be the same or they can be different. If the subunits are the same, then the protein is known as oligomeric. Oligomeric. Same subunits. So for example, so we'll talk about hemoglobin in chapter 5. So this protein, it's a transport protein, transports oxygen through the blood. It has 4 subunits. Now, 2 of them are so there are actually comes in 2 pairs. There are 2 alpha subunits and 2 beta subunits. So it's not fully oligomeric protein. It's a 4 subunit protein, not fully oligomeric because alpha is not the same as beta, but it's composed of 2 oligomeric parts, right? So 4 subunits in total. So one other thing to consider, any questions, by the way? Any questions at this point?
I have a question.
Sure.
So the multi-subunit, would that just be primarily for the one that is by the bovine, or does that go for everyone? Does that make sense? Like, which ones have like the multi-subunits?
Oh, so this is the number of chains. So basically anything than 1 will be multi-subunit.
Okay, thank you.
Right. I just wrote it here. It doesn't mean it's this one. It's so multi-subunit will be this one, that one. This has 12 subunits, right? Glutamine synthetase. So now the question is, can you tell if you have a molecular weight, can you tell how many residues, amino acid residues you have in a protein based on the molecular weight? So let's say I have molecular weight is 12,400. can I, and I don't know how many amino acids, right? So for example, I obtained protein for the first time. Nobody has ever isolated this protein before. I use mass spectrometry, as we will see later, to identify the molecular weight. Can I tell how many amino acids my protein has? What do you think?
Wasn't there a way to estimate it by, it was like something that you could divide the molecular weight by? I can't remember. I think it might have been like 113.
Yeah, very close. So very good point. Now, so there are 20 amino acids. I mean, it would be ideal if each of these 20 amino acids had the same molecular weight. Then not only we could estimate, we could precisely say how many amino acids we have, right? The problem is that proteins vary in composition, and the 20 amino acids have different molecular weights. But if you look, interesting, you said you have to divide by a certain number. The interesting thing is that the average, okay, let me go to the empty page. So this will be an empty page, and so the average mole weight of 20 amino acids is 138. So one way to do that would be to take the molecular weight of this protein and divide it by 138, and hopefully you can get a good estimate of the number of residues. But look at something peculiar here. So look at the differences between this protein and that protein. So the difference in molecular weight is only about like 1,000, slightly more than 1,000, right? But the difference in the number of residues is quite significant. There are 20 more amino acids here. Why do you think that is? So obviously it's not just, there's something about this average molecular weight of 138, so which is not exactly a good number for us to use. So why do you think the differences are not proportional?
Is it because it's using like more amino acids that are heavier?
Which one? Which one uses heavy amino acids?
The ribonuclease?
Well, I would think they're lighter, don't you think? So basically you have more amino acids in this protein than in that protein, even though the molecular weight is similar. So which means the ribonuclease uses more lighter amino acids, correct? And in general, most proteins out of 20 amino acids, they utilize lighter amino acids. And so better number to use for the average weight is 128. Even though this is not the average weight of the 20 amino acids, but this takes into account that most proteins have more lighter amino acids than heavier amino acids. Again, because proteins have more, take a long time to write, have more lighter. Is this clear so far? Yes? more lighter amino acids. Now the killer question, which should wake you up. I know you just woke up anyway, but this should wake you up completely. Now, do I take, now with this one number 128, do I take this number, or 12, for example, 12,400, divide by 128, will I get something close to 104? What do you think?
Yes, it's pretty close. It's just a little bit more.
Oh, you already calculated? Okay. So, well, I was expecting a different answer from you. And so it should not be very close. Well, it could be close, but it could be much closer if we took into account that proteins are not just mixtures of amino acids, right? So let's look at this slide before. What happens when we make a protein? We have these amino acids, right? So proteins are not these mixtures. The proteins are amides, which means in the process of their formation, amino acids will lose water, right? So then we'll go back to this number and we have to subtract the molecular weight of water for each amino acid. So do you understand what I'm saying? Need to subtract subtract 18. So the average weight, so the number to use is 110. Okay, so that will give you the best estimation of the number of amino acids in a protein of a particular molecular weight. Is this discussion clear, guys? Yes?
Yes, that makes sense.
Yep.
Yeah, okay. So first we go from the average weight of amino acid, we account for the fact that the lighter amino acids are more prevalent in proteins, and then we subtract water from each amino acid having to be coupled to the next one, which is 18, and you get 128 minus 18, you get 110. All right, so that's the number you use on the sapling and on the enzyme. Sorry, enzyme, on the exam. All right. And lastly, as far as proteins, the proteins can also be bound to other large molecules, right? The proteins can be bound to lipids. We will actually see those lipoproteins. The proteins can be bound to carbohydrates, glycoproteins. In fact, most proteins, for example, on cell surface, on any kind of cell surface, are glycoproteins. Proteins are virtually decorated with various sugar residues. And actually, this one way, for example, there's one particular one, good to remember. We will look at this when we talk about carbohydrates, sialic acid. Sialic acid, it's a type of carbohydrate which is attached to protein on the cell surface and particularly overexpressed by cancer cells. So when we talk about, you will see that I keep coming back to this issue of selectivity between inhibiting the growth of cancer cells versus normal cells and reducing the side effect of chemotherapy. Taking advantage of, for example, of the fact that cancer cells have different glycoproteins on their cell surface. So sialic acid is one of the reasons for that. We can selectively target cancer cells over normal cells. Then there are phosphoproteins, right? So many amino acids will have phosphates, hemoproteins, heme, flavoproteins, different nucleotides, metalloproteins. So we talked about those as well. All right. So when we deal with proteins, so for the rest of this class, right, we just basically just started. We're just on chapter 3. What we're going to be covering are these questions, and so we're going to be learning how to deal with these questions, how to answer them, and which techniques to use. So today we'll look at the, for example, how to determine sequence, right? So that's number one question you ask. Okay, you have a protein. Okay, 100 amino acids. The question is, what are these amino acids? That's the first question you ask. Right, and what else is there? Is there a component? Is there a metal there? Is there cofactor, right? And then the next chapter will deal with three-dimensional structure, right? So the proteins are large, and they will undergo specific folding, and it's the folding of the protein which will determine its function, right? This three-dimensional structure is crucially important to the function of the protein. So no, there are no proteins that I know of that are simply stretched into like a string in a one-dimensional direction. Maybe there are, but I just don't know. Would be very uncommon. So most proteins are folded into a particular three-dimensional shape. How does it find a native fold? So we'll talk about that as well. All right, so if it's folded, how does it fold? How does it know how to fold, right? So it's not just a simple random folding into a ball, right? From a string to a ball. No, it's a specific three-dimensional structure. So the question is, how does it find it? Then once it's folded, how does the chip?
I have a question. Are we going to talk about prions in this class?
Prions, yes, we'll touch on prions, yes. Mm-hmm.
Thank you.
Yes. We'll touch on prions. So the question is, how does it achieve its biochemical role, right? So if it's an enzyme, how does it work, right? How does it perform a specific chemistry? And then how does this reaction stop? How does this reaction accelerate it, right? What other molecules are important and which sites on the enzyme are important for regulation? And how does it interact with other macromolecules, right? If it's bound to lipid, bound to carbohydrate, or how does it interact with other proteins, right? So there are a lot of protein-protein interactions in cells. So proteins interact with other proteins. In fact, it's a huge area in drug discovery right now, is to disrupt protein-protein interactions. Protein-protein interaction. So that if the two proteins interact and there is a specific function for that interaction, how can we disrupt it if this function leads to a disease? Not very easy, because proteins are large molecules. And if your drug is a small molecule, it's very hard to disrupt the protein-protein interaction with a small molecule. Just imagine proteins will have large surfaces, right? Large surface areas. Let's say my 2 palms are my 2 proteins, right? And then I have a penny, which is a drug, and I put, and my 2 palms, let's say I have some kind of glue, right, on my palms. And if I bring them together, they'll be glued together, right, because of the glue on both palms. And let's say I put a penny on one of the palms, hoping to prevent my palms coming together and getting glued to each other. The penny will not do a good job, right? It's a very small object. So a huge problem in drug discovery is how to develop such small molecules that can disrupt such protein-protein interaction. Okay, how is it related to other proteins? I guess this will be if you know the structure of one protein and you know that there's similarity in function, maybe there's similarity in structure. Where is it localized within cell, right? Does it work in the mitochondria, in the energy organelle? Does it work in the ER, in the plasmic reticulum, in the cytoplasm? Depending on where it is, it performs a different function. And what are its physicochemical properties? Right, so these would be important for what we're going to discuss just now. How to purify and separate proteins. If you have a mixture, and usually, let's say you get a protein out of a cell, you need to purify it from a mixture. There's a huge number of proteins in a cell. How do you get one specific protein out of that mix? And so the chemical character, so we'll take advantage of a number of differences. We'll talk about the sequence and arrangements of amino acids, right? So there will be different chemical characters. So there will be amino acids which are charged, polar, hydrophobic, and so on and so forth. Some proteins have more hydrophobic than others. So we're going to try to separate them based on that. Some of them are more polar. Some of them are more positively charged. Some of them are more negatively charged. So we'll take advantage of those properties to separate proteins. And other times it's possible to what's known as go fishing, kind of fish them out. So for example, if you know there's a specific target, for example, there's a small molecule that binds to a particular protein. You can use this small molecule as a bait. Like you go fishing, you put a bait, right, on your hook. You put a bait and you throw it in the water. And fish will bite it. And so the same idea could be used to fish out proteins from a mixture. You basically have this small molecule ligand that will bind to your protein, but not to other proteins, right? You attach it to something, let's say to some kind of solid support. Usually this is some kind of beads or some kind of particles, large particles, and expose your protein mixture to these particles. And so the proteins which have affinity to this target, this ligand, right? They will bind to your whatever particle it is, and then you just wash the particles off. All the non-specifically bound proteins will come off, and you only have the proteins that have taken the bait, right? That have attached themselves to the ligand. Very fruitful area, and a lot of great examples of purifying proteins based on using these specific targets for fishing. And so separation will be based on physical and chemical properties. So charge, we just talked about. Size, right? So you can separate proteins based on their size. We just talked about affinity for a ligand. Solubility, right? So some proteins can be soluble in water more so than others, and you can take advantage of that. Hydrophobicity, thermal stability. For example, one of the common techniques for solubility is known as salting out. Salting out. You probably know that from general chemistry. Have you heard of this term, ionic strength? Ionic strength. It's crucial for understanding the salting out technique. Ionic strength. So we know that proteins, so those proteins that live, for example, in the cytoplasm, right, they're water soluble. But some of them more water soluble than others, right? But water, it's not just pure water. It's, let's say water can have lots of different salts in it. And as you increase the amount of salt in the water, you will increase the ionic strength of water. And the water will become more polar. More polar. So if your protein, for example, is not sufficiently polar, and you make the water more polar, you have a mismatch. Remember, like dissolves like, right? So the polarity of the protein has to match the polarity of water. And if you can vary, you can change the polarity of water by adding salt, by changing the ionic strength, you can change the solubility of different proteins. And so what you can do, usually different salts are used, and ammonium sulfate is one of them. NH4. SO twice SO4, ammonium sulfate. It's a salt which is commonly used for salting out proteins, right? So you add the salt, you change the ionic strength of water, and the proteins which are not soluble in this new media will precipitate. And then you just collect them by centrifugation or filtration. And so salting out is probably one of the first things you can do to get the desired protein out of a mixture. It will not give you a pure single protein. Usually differences in solubilities are minor. And so, but it will, I don't know, if you have 100 different proteins, maybe you can get 10 out of 100. So it's already pretty good. You got rid of 90% of junk, right? Now, size, very important as well. There is a technique known as dialysis. Do you guys know what dialysis is? What's dialysis?
It gets rid of toxins in the blood.
So how is it done? What do you use? Like what is it based on? So I just mentioned to you size. Obviously the size difference has to be huge. So you cannot separate one protein from another using this technique, but you can separate proteins from small molecules very easily. So as I said, toxins, small molecules, right? So you can keep all the cells and proteins in the blood, but get rid of all the toxins, for example, right? But basically what happens in dialysis, basically you put your protein in a bag which has a semi-permeable membrane through which the protein will not pass, but small molecules can pass. And then you submerge this bag into some kind of buffer which will not kill the three-dimensional structure of the protein. Be careful about using just pure water, right? So pure water is rarely used with proteins because pure water can break up all sorts of hydrogen bonds within the protein and make it misfold, right? So folding is very highly dependent on the pH and of the media. So you will put the protein in this semi bag with a semi-permeable membrane, and then submerge it in the buffer. And so the small molecules which were mixed together with the protein, because of the concentration gradient, right, they will pass out of the bag into this solution of the buffer. And that way you can get rid of the bulk of the small molecules. There'll still be an equilibrium established, but you can do dialysis multiple times, right? You do it once, you get rid of some portion of the small molecule, then you get rid of that solution, the buffer, put it in next buffer solution, and so on and so forth, until you bring the concentration of the small molecule to a minimum. All right, so these are some of the basic techniques you can use. Again, these will not give you complete pure proteins, but these are some of the first things you can do to start purifying the protein. All right, chromatography. Chromatography, very useful technique. So usually there is some kind of stationary phase used. So there's a column. Now you're familiar with column chromatography from even, for example, from taking organic chemistry 2, right? Or 1 lab. So there'll be some kind of solvent, right? Some kind of solvent. So you will apply the protein sample on top of the column, right? So there's a stationary phase which has been soaking in the mobile phase, right? And so there's a porous support here through which the solution will pass through, but the stationary phase will not pass through. And so there's a pump, and you start running the solvent through the column. So this is time 0. After a certain period of time, you can see different proteins. They even have some colors to them, which is nice. Not always you get color, but if the proteins are colored, the purification is even easier. So you can see there's a band. There are different bands, right? So the proteins start to be separating based on the interaction with this solid support. So basically, the separation is based on equilibrium, which is established for each protein. So the protein can spend some time in a solution and can spend some time adsorbed to the solid support. If it spends a lot of time in a solution, it will pass through very quickly, like this, right? Because the solvent will push it through. If it spends a lot of time being attached to the solid support, it will migrate very slowly. And so here you can split it into fractions A, B, C. There's some kind of detector, so you can use some kind of property, for example, absorption of the UV light, right? And you can detect various fractions using that. So this is column chromatography. This is the most common one. Now, I told you that the proteins can be separated by charge, right? So there are a lot of ionizable amino acids present in proteins. Some of them have more negative, some of them have more positive. So here we're gonna use some kind of resin. So resin is usually a polymer that has charged groups. So in this case, for this separation, we're gonna use resin which is negatively charged. So if the resin is negatively charged, it's known as a cation exchange resin. Okay, so cation exchanger. So it's the opposite. So this is how I remember this. So if the resin has a negative charge, it will be exchanging cations. So it's a cation exchanger. If the resin is positive, it will be an anion exchanging resin. And so what happens here is that if your protein, which is now the small sphere, right? If it has a lot of positive charge, such as this, blue, right? Has a lot of positive charge. It will be interacting with the resin and will not go through the column easily. Now this one, now I'm a little colorblind, but this is red, right? Is this red?
Yes.
Red, yeah. So the red one, which has large negative charge, it will be repelled, not even attracted. It will be repelled by the resin and will spend most of its time in solution, and the solvent will just take it through the column very quickly, right? And so depending on the charge of your protein, you can separate proteins into various fractions, right? So here's your first example that will definitely be on the test. Test question, another test question. Another test question. So I have a protein that has a lot of aspartate amino acids, and I have a protein that has a lot of lysine amino acids. Which protein will elute first in this column? So aspartate and lysine.
Aspartate,
Why?
because it has the negative charge and I think the lysine has a positive charge.
Yes. Right. And so based on our prior discussion, so aspartate has a large negative charge. It will be repelled by the resin. Yes, that's what your reasoning was. And the aspartate will come out first. Right, so I will basically, what I will tell you, for example, on a test, different proteins, I'll give you amino acid composition. You will need to predict the charge of the, maybe peptide, not a protein, a peptide. So that's easier to handle. Predict the charge, basically add up the charges, find the total charge of the protein, and it will tell you if it's, and compare the total charges between different proteins. And depending on that, also keep in mind of which kind of resin it is. If it's a cation exchange resin, then the result is as shown here. If it's an anion exchange resin, then the result will be the opposite. Cation exchangers versus anion. So don't get confused. Cation exchange resin is not the resin that has positive charges. It's the resin that exchanges positive charges, right? So the resin itself is negatively charged. This is cation exchange. This is cation exchange versus anion exchange. So I used to get confused about this myself in the past. Versus anion exchange. And this will do the opposite, right? All right, separation by charge. So size exclusion. So dialysis works for proteins versus really small molecules, but if the difference in sizes is not so great, if you're dealing with two different proteins, one, let's say, is half the size of the other, dialysis will not work as well. So in this case, you will do size exclusion chromatography. Here you have some kind of polymer beads, which will have pores in them, right? And so small proteins can get stuck in these pores. They can get stuck in these pores and spend some time in these pores and migrate through these pores more slowly than they would migrate through the in between these beads, right? And so the small protein will migrate slowly, large protein will migrate quickly, right? And so the large protein will come out in the earlier fractions, and the small proteins will come out in later fractions. So size exclusion is actually pretty straightforward. Just keep in mind that the result is counterintuitive, right? So you would predict that normally when somebody asks you, I have small protein and large protein, which one will come out first? So the first answer without thinking would be small protein, of course. But it's actually the other way around, right? So the small protein will get stuck in the pores, and the large protein will just bypass them because it will not be able to get inside the pores. And so here's our binding affinity. So we talked about that, right? So it's a little more complex representation, but the idea is the same. So here's our bead, and we are attaching a ligand to our polymer bead. And so the protein of interest will have a site that will have affinity for the ligand, right? And so if you put these beads into the column that have the ligand attached to them, then the protein that have these sites, the site here, right, the site there, sites which are capable of interacting with the ligand, have some kind of binding affinity for it, they'll get stuck. They'll get stuck, and all the other proteins that do not have this binding affinity will pass right through. You'll collect those, and then the question is, how do you release these prisoners, right? How do you release these proteins which are now stuck in a column because they're attached to the ligand? The easiest way to do that would be to actually add the ligand to the column by itself. That's what I would do. Just basically add the ligand by itself into the column. The ligand will compete with the stationary phase. So you have ligands in two places. There'll be ligand on the stationary phase, which we used originally, and there'll be ligand that is in solution, right? So they will both compete for the protein of interest. And so the protein of interest will come off the stationary phase and bind to this soluble ligand. And now it's no longer attached to the stationary phase. It can pass right through. So at the end, you still have a protein attached to the ligand. So you still have to solve that problem, but you can do it by dialysis, for example, right? So you can separate those by dialysis. So this is binding affinity. Electrophoresis. Okay, so far we've talked about preparative scale, right? So you want to isolate a protein from a mixture, and that's usually done on large scale. If you want to have milligrams of proteins or even grams of proteins. Now, how about analytical scale, right? So you don't have much sample, and you just want to be able to analyze what proteins are present in your mixture. You don't want to isolate one specifically to, I'm sorry, my phone ringing, it's pretty far. So you don't want to isolate the protein to actually, to do some kind of reactions with it. You just want to be able to detect it, for example, in a mixture, right? And so in this case, you will use electrophoresis. And here, the main components of an electrophoretic apparatus will be the electric field, right? So the proteins will migrate according to their charge. You will have the gel matrix, which will hinder the mobility of proteins, right? So the electric field will pull the proteins based on their charge. But the proteins will not be pulled so easily because there'll be gel matrix. So this will be a cross-linked polymer, polyacrylamide, for example. That will make it difficult for large molecules to diffuse through, okay? And so the gel is commonly polyacrylamide, so separation of proteins is often called polyacrylamide gel electrophoresis, or PAGE. So PAGE, if you see PAGE, this is what it stands for. Polyacrylamide gel electrophoresis. This is something which is not in the book, but something that you can think about. This is acrylamide. How do you make a polymer out of this? Yeah. How do you make a polymer out of it? This should add to the amount of knowledge you gain from this class. Not only you know how to use gel electrophoresis, you will know actually what stationary phase, what the gel looks like, from the chemical perspective. So what you will do is there are these wells at the top, right? So there will be the apparatus itself, which is placed in the electric field, right? So this is positive, this is negative, and so you place the protein sample into these wells, right, and apply the electric field to them. And so the proteins will migrate. So for example, the negatively charged proteins will migrate in this direction, right, from the towards the anode, towards the positively charged electrode.
Is this how like certain base pairs of DNA are separated as well?
Yeah, this is also can be used to separate DNA. Yes, and we will talk about DNA separation, various techniques to separate DNA as well. Yeah, when we talk about DNA. Mm-hmm. But this is a very common technique for separating large macromolecules based on their size and charge. Right. So here, what will happen is that these small molecules, the smaller proteins, will migrate faster because of the diffusion, right? So because polyacrylamide is a cross-linked stationary phase, which will inhibit the migration of large molecules. So the large molecules will get entangled in the polyacrylamide chains. And so it's going to be very hard for them to break through. It's almost like, imagine like if you go through a jungle, right? You go through a jungle and there are all these branches and so on and so forth, and try to go through a jungle. If you spread your arms, for example, and you're a large person, you spread your arms, right? As opposed to if you're a small person, you just go straight through in between the branches. So same idea with gel electrophoresis. Oftentimes what is done is the technique is not just PAGE. Technique is also known as SDS PAGE, which is an improved version of that. And the reason for why it's improved version of it, so if we want to separate proteins based on their size, unfortunately, we will also be separating proteins based on their shape, just like I told you, right? So proteins which get entangled more easily, they will have lots of branches by themselves, right? But if you can unfold the protein into a linear protein, for example, that does not have a three-dimensional shape, then you'll be separating all these proteins based purely on their size, not on their shape. And that's what the sodium dodecyl sulfate does, right? Basically, this hydrophobic chain, what it does, it incorporates inside the protein, it sticks its tail into a protein and breaks up any kind of hydrophobic contacts between the amino acids within the fold, within the folded protein. And so the protein basically unfolds. And now also you add a negative charge to the protein, right? So the native charge doesn't matter anymore. So you unfold. So native shape does not matter. Using this tail for unfolding. And the charge, negative charge, will give the protein uniformly negative charge. So I'll give this 2 stars. That's 1 star, and that's 2 stars. So it accomplishes 2 functions, unfolding and the charge. And so the proteins will migrate towards the positive electrode based solely on their size, right? Small proteins will move faster. So SDS page is now a very common technique in most biochemistry labs because it's so efficient. But keep in mind, it's only on analytical scale, right? But analytical scale is very useful oftentimes. So this is a picture basically of a standard of a gel with the protein bands that has been obtained after the electrophoresis procedure. And you can see here there are markers. So this is a mixture of various proteins that you can apply to know the migration rate, right? So you know, for example, whatever protein migrates up to here or down to here, right, will have a molecular weight of about 14,000. Whatever protein migrates very little, just this much, will have molecular weight close to 100,000, right? So these are molecular weight markers, which will give you an approximate weight of your protein bands and your protein sample that you separated by SDS page. And for example, you're looking for a protein, this guy, right? And you know that you can, we will actually learn later in this class, but you will also learn in biology classes, you can induce an expression of particular proteins in any cell, right? So cells don't always make all the proteins at the same time, right? Depending on the circumstances. For example, if a cell divides, it produces more proteins which are required for cell division, right? If the cell is in resting state, it will not produce these proteins. So, but you can artificially induce the production of particular protein in a cell. So here, this will be a cell where the this protein is not induced and it's absent, right? So the protein, for example, molecular weight 45,000 is absent. And these cells are induced. So the protein was induced, and you can see there's quite a bit of it that has been expressed. Now, this is soluble crude extract. So this is after you break up the cell. You break up the cell and open it to and release all the proteins. So now there's much more of this protein, much higher concentration of all these proteins. And there's tons of this protein that you're looking for. And then you do the salting out technique that I was telling you about. And you can actually get a pretty good purification using this technique here. There is some garbage here a little bit, but for the most part, it's quite pure. And then you know that your protein has partial negative charge, for example, right? So you will use a what do we call it? What did we call the? Sorry, I'm getting tired. Sorry, ion exchange chromatography, right? So this particular one is anion exchange. So you will use the positively charged beads, and so your protein has partial negative charge somewhere. So you can see this purification actually gives you a much cleaner protein this time. Now you can apply it to cation exchange to get rid of maybe of some proteins that have the opposite charge. Also gives you quite a pure protein. And finally, after all this, you get a nice pure protein, right? So this is the series of steps you take to purify this protein. But again, this is analytical scale, and you cannot really use it to produce protein in large quantities.
So for the ion exchange, would you use both the anion and cation exchange like in succession, or would you use one or the other?
Well, I guess you can use both. I don't know if it matters which one you use first or second. Basically, in one of them, your protein will migrate slowly. In the other, your protein will migrate quickly. So if the protein migrates slowly, you will get rid of all the proteins which migrate quickly, right? They will come out in earlier fractions. If your protein migrates quickly, you will leave all these other proteins behind because they migrate slowly. So it's kind of you're looking at both ends of the spectrum, right? So you get rid of using both of these, you get rid of proteins which are negatively charged, and you get rid of proteins which are positively charged. So I guess it improves the separation because you use both, and not just focus on one particular type of proteins. Does it make sense?
Yes, sir. Thank you.
Yeah. All right. And so obviously with SDS page, you can use to calculate the molecular weight of the protein. The technique is analytical, very precise. So again, this is the molecular marker, right, which will give you different proteins. Now, by the way, I have to tell you that you can stain these proteins with various dyes. And one which is most commonly used is known as Coomassie blue. Coomassie blue. No, I don't know how to spell that. I think something like that, or close. Coomassie blue. That's why you see a lot of these bands in blue color. So this dye specifically binds to proteins, but doesn't bind to the polymer, to the polyacrylamide. And you can visualize proteins with the polyacrylamide on the background. So you can calculate the molecular weight of the protein. So here you can have a plot, right? You find your protein on this plot based on the relative migration, and you can calculate the molecular weight of the protein. All right, and the last technique I want to show you is what's known as isoelectric focusing. So we talked about isoelectric point, the PI, right? Point of equivalence. That's when the protein has no charge. What we talked about in terms of amino acids having no charge, being zwitterions, right? So, but the protein can also have no charge at a particular pH value. And that would be the case when all the negative charge, the sum of the negative charges equals the sum of the positive charges. And so what you will do is you have this a strip, which is which can be filled with small molecule acids and bases, and you can place this this gel strip, I think some kind of polymeric gel strip, you can put it in the electric field. And so these small molecules, they're known as amphilites, small molecules, acids and bases. They will basically they will localize based on their own pI values, right? And create a certain pH, because their pIs will equal the pH values in the particular origin of the strip, right? And so what will happen as you apply the electric field, the protein will migrate. The protein will migrate, let's say from, and you start with pH 9, high pH, to pH low 3. And so the proteins will migrate to the point of their equivalence and stop right there. And they will stop because they become neutral. They're no longer attracted to the positively charged electrode, and they're not attracted to the negatively charged electrode either. So they'll just stay there. And so you can separate proteins based on the differences in their pI values. So this is known as isoelectric focusing, right? And so so useful technique, and it's even much more useful if you can combine it with SDS page. So this is known as two-dimensional electrophoresis. Now, when you're dealing with hundreds or maybe even thousands of proteins, very useful to be able to separate proteins based on this two-dimensional technique. So what you will do is you'll do the isoelectric focusing first, right? You have this strip with various proteins, maybe some here, some there, along the strips, right? Separated based on their differences in their pIs. And then you apply this strip to the electrophoresis gel, which will make the proteins migrate. All these proteins will migrate down through the polyacrylamide gel. And what will happen, you will have proteins separated horizontally, right, based on their isoelectric focusing, and you'll have the proteins separated vertically based on the differences in their molecular weight. So basically, it's the whole gel will be filled with proteins, not just on one line, but in two dimensions. Differences in pI, differences in molecular weight. All right, and I already told you that about the light absorption. So you can also visualize proteins using UV light. That's another way to visualize. So using different stains is one. UV light is different. And this is primarily, remember, the two amino acids, tryptophan and tyrosine. I showed this to you earlier, that tryptophan absorbs quite a bit of ultraviolet light at 280, right? This is absorbance. This is nanometers of light, and tyrosine absorbs quite a bit. And since most proteins absorb light, most proteins have tryptophan and tyrosine, or a number of those, they will absorb UV light. Obviously, those that don't will poorly absorb. Like polyglycine, for example, that only has glycine, will absorb light very poorly. Okay, so we still have a couple of slides to go through, which we don't have time for. So I will assign reading for you to do. And I will put it in the announcement of the Canvas, okay? And if you have any questions, guys, about the reading from those pages that I assign, please just put them in the discussion of Canvas, okay? So you can obviously email me, but it's much less productive for the class because I would like everybody to participate and everybody learn from our discussions. Are there any questions about today's lecture? Anything that was not clear? Anyway, if it wasn't, Canvas, go to Canvas and write it in there. All right, the homework is due Monday, and on Monday we start chapter 4. Stay safe.
Thank you.
Bye.
Thank you.